My name is Yasuo Kurimoto from Kobe, Kobe City Medical Center General Hospital, Department of Ophthalmology.
Dr. Kurimoto, could you tell us about your keynote presentation made at AAO 2015?
I made a presentation about IPS project, which was IPS-derived retinal pigment epithelium transplantation for exudative AMD. I presented in the retinal subspecialty day.
What were the key findings?
Okay, so iPS cell has the same ability as ES cells have, but iPS cell have several advantages as compared with ES cell. Most important advantage is it's free from ethical issues because iPS is derived from autologous somatic cell. So as the donor, only you need is just a small piece of skin, and also no immune problem because it's autograft. So we transplanted the RP, IPS-derived RPE to the AMD patient, and current our stage is to confirm the safety. So we just finished 1 year's follow-up of the patient, and we confirmed the safety of the IPS cell as a donor.
What would you say was the key innovation or the difference in your approach for this study versus what other people have done with iPS cells?
We succeeded in making RP cell sheet without any artificial material. So, and we succeeded in transplanting RP sheet in subretinal space in AMD patient. Both are pretty difficult because RP sheet without artificial material is very difficult to treat, but we succeeded in that. And also, people wondered, so maybe RPS could be differentiated into cancer. So we checked very carefully. So we made immunogenicity test, and we checked whole genome analysis. And about the first case, so our RP sheet cleared all such issues.
What was the duration of the study?
Duration of
how long did you follow these iPS cells?
Okay, so 4 years. So we just finished 1 year, and we will follow more 3 years.
What are the next steps for your research in this area?
Yeah, so actually now Japanese government is changing the regulation, so we are waiting the result of government review board. So because anybody know what is the safety limitation, what is the safety guideline about the iPS cell. So because some mutation of DNA is inevitable. So but anybody know what mutation is safe, but what and how much mutation is dangerous. So now our government review board are addressing that issue. So we are waiting their result, their conclusion.
Okay. Tell us a little bit about some of the other research that you're doing in your department.
My department?
Yeah.
Okay, so about iPS cells. Okay, so it's in the stage of animal experiment, but we are researching in photoreceptor sheet. So we succeeded in making photoreceptor sheet in mouse. So maybe the photoreceptor sheet transplantation will be the next clinical project of us.
If you look at your current research, what is the timeline to seeing this in the clinic? What are the translational timeline?
Okay, so So you mean daily practice, clinical practice?
Right.
Okay, so maybe we need more 10 years. But I think the first target is non-responder to anti-VEGF therapy. So if the issue of cost is solved, anytime we can apply RPC to such non-responder to anti-VEGF therapy.
So if you were to summarize the key hurdles or the key problems, key challenges that you still face, what would those be?
Key challenge about iPS
in
cell
terms
project.
in terms of getting iPS cells that remain viable, that continue to be healthy in this particular environment and will yield clinical outcomes.
So
What are the major problems that you face from a research perspective?
Okay, so I think three major problems. One is cost, I told, and second is safety about gene mutation, and the last problem is safety of surgery, because as you know, subretinal transplantation is associated with some invasion. So it's not like cataract surgery. So how we can make the surgery more safer is one of the main problems.
Professor, thank you so much for
Thank
visiting
you.
with us. We really appreciate
You're welcome.
it. And we'll follow your research closely.
Thank you.
Thank you so much.